Hi, everyone. In the last lecture, we explored how microscopes revealed an invisible world of microbial life. But how do microscopes actually work? In this lecture, we'll look at the basic principles behind light microscopy, including how microscopes use light and lenses to magnify tiny objects, why microscopes have limits, and how scientists improve image quality to study microorganisms. By the end of this module, you should be able to explain how light microscopes magnify and resolve microscopic objects, compare major types of light microscopy used in and compare major types of light microscopy used in microbiology. Light microscopes allow humans to observe structures far too small to see with the naked eye. They work by using visible light along with a series of lenses that magnify tiny objects. This allows scientists to observe cells, tissues, and microorganisms that would otherwise be invisible. But magnification alone is not enough. To understand how microscopes produce useful images, we first need to understand one important property of light itself. Light behaves as a wave. Like other waves, light has a wavelength, the distance between one wave peak and the next. Different wavelengths carry different amounts of detail. Shorter wavelengths can distinguish smaller distances between objects, which means they provide higher resolution. This is important because the wavelength of light ultimately limits how small an object can be for a light microscope to clearly resolve it. Visible light is only a very small portion of the electromagnetic spectrum. Different wavelengths within visible light correspond to different colors. Light microscopes specifically use visible wavelengths of light to generate images. Because visible light has relatively large wavelengths compared to molecules and viruses, light microscopes eventually reach a physical limit to their resolving power. This is why some structures are simply too small to be clearly visualized using standard light microscopy. And just to orient you to this screen here, on the left-hand side, as we get into smaller and smaller wavelengths, we get into the ultraviolet X-rays, and then we get into like crazy radiation, and like the smallest would be like a single electron. And on the bigger end, we get into like infrared, and then we get into some other wavelengths that we don't really use for visualizing too much, but they have to do with like electricity and stuff like that, and radio waves. A compound light microscope contains several important components that work together to form an image. The ocular lens, which is up there at the top with the eyepiece, um, is magnifies the image viewed by the observer. The objective lenses provide most of the magnification and can be switched depending on the desired level of detail. And so those objective lenses are the ones that kind of flip around in a circle that you see at point number 3 there. The condenser focuses the light on the specimen, helping to improve the image quality. And so we can see the condenser there around number 8 on the image. And the stage holds the slide in position. So the stage is there at number 6. And while so the stage holds it in position while the specimen is being viewed. Together, these components manipulate light to produce a magnified image of the sample. Magnification describes how much larger an object appears compared to its actual size. In a compound microscope, total magnification is determined by multiplying the ocular lens magnification by the objective lens magnification. For example, if the ocular lens is a 10x lens, that means 10 times, and we have an objective lens of 100x, what we do is we multiply 10x times 100x, and we get a total magnification of 1000x. Um, However, increasing magnification does not automatically improve image quality. A blurry image that is enlarged is still blurry. That's why resolution is just as important as magnification. Now, for purposes of knowing material for the exam, you should be able to do a lens total magnification calculation. And the way you do that is simply taking all the lenses that are in series and multiplying by each other, right? So if you have objective lens of 4x and an ocular lens of 10x, it's 4 times 10. The x is not going to like get squared or anything because it's not an actual unit or unknown. So we just say 4x times 10x equals 40x. All right. Now, resolution refers to the ability to distinguish two nearby objects as separate from one another. Higher resolution reveals finer detail and produces clearer images. The resolving power of a light microscope is limited by the wavelength of visible light. So most light microscopes can resolve structures down to about zero point 0.2 micrometers. That's about 200 nanometers. Below that limit, nearby objects begin to blur together and can no longer be distinguished clearly. This is one reason why viruses are difficult or impossible to observe using standard light microscopy. One way scientists improve resolution is through the use of immersion oil. Normally, some light scatters as it moves from the glass slide through the air and into the objective lens. Immersion oil reduces this scattering because it has optical properties similar to glass. As a result, more light enters the objective lens, improving image clarity and resolution. Oil immersion is commonly used with a 100 times objective lens when observing bacteria and other very small cells. And it's not used with any of the other lenses. So if you have a 40x lens, which is called our high dry lens, we never want to put oil on that. The oil can get underneath the lens and permanently damage those lenses. They are quite expensive to replace. Even with good magnification and resolution, many microorganisms are still difficult to see because they are nearly transparent. Contrast helps make cells and structures more visible. One common way to increase contrast is through staining. Stains are chemicals that bind to cells or cellular structures, making them easier to observe under a microscope. On this slide, we have a pair of unstained cells on the left and stained cells on the right. Different stains can highlight different features of microorganisms. So if you look here on the left-hand side, it might just look like a gray blob, but if you kind of like focus in, you can see there's sort of an outline of a cell. That's what cells look like when we don't stain them usually. It's very hard to see them unless they are quite big. They kind of bleach out because the light passing through them produces what's called a photo bleaching effect. And so you don't really see them because they're kind of colorless to begin with. But on the right-hand side, you see when we add a little bit of stain, in this case, I think it's crystal violet, you see that you have very clear, easy to see purple cells. Later in this course, we'll explore staining techniques in much greater detail. Light microscopes transformed biology by allowing scientists to observe structures far too small for the naked eye. But not all microscopes work in the same way. Different microscopy techniques are designed to solve different problems, improving contrast, visualizing living cells, or revealing specific cellular structures. In the next lecture, we'll explore the major types of microscopy and how each technique provides a different view of the microbial world. Thank you for attending, and see you next time.